and we're going to kick off our Molecules to Medicine, Making Vaccines in a Global Pandemic session for the BT Young Scientist. So I'm just going to introduce myself really briefly. My name is Sarah and I work for SSPC. And we're a Science Foundation Ireland Research Centre for Pharmaceuticals. And what we try to do is we try to bring Irish research expertise, developing innovative technologies to address key challenges facing the pharma and the biopharmaceutical industry. So in English, we bring academics from all the different universities, all the guys in the pharma industry together, and we try and come up with solutions to problems that they're facing and make medicines in a better way. So I'm only just back from maternity leave, and I don't know, I was having all these side conversations with people about COVID, about vaccines, about all the different challenges that we were facing. And I was getting so many questions. You know, when you have a science background, you get so many questions from people who think you have all the answers and all the solutions. And I absolutely, most definitely didn't. And it got me to thinking, God, it would be really great if we could convene a panel of people who work in this area, who work on making medicines and making vaccines, to discuss some of the challenges and maybe answer some of the questions. And that's how this panel came about. And we thought the BT Young Scientist was such a fabulous platform to do it, because young people have been so disproportionately affected really with this whole pandemic, you know, missing out on the really important things like social life and schools. And we wanted to offer a platform for young people, their parents, their teachers, to be able to engage with us and ask questions. So that's a little bit of background to this panel discussion. And I'd like to say a really big thank you to all of our panelists. And I'm going to hand over to Luke now, and I'll be back with some questions later.
Great, thanks very much, Sarah. Very happy to take part, of course. So many questions have come in already, haven't they? So we could be here for a while, I guess. But I'm very happy to be joined by the fellow panelists here. So we're going to mainly talk about, I guess, vaccines, but also therapeutics, which is a big focus for many people on this session as well. So, well, we might start by introducing each of us. So I might start with myself. So I'm Luke O'Neill. I'm the Professor of Biochemistry in Trinity. I'm an immunologist. I work on COVID. I've got 4 projects in my lab at the moment. We work in the therapeutic end of it, actually, mainly developing anti-inflammatories to treat some of the symptomology of the disease. So that's what my main focus in terms of COVID is, as well as trying to keep the public informed, I guess. So we could move on. Sarah Hudson, do you want to introduce yourself?
So I'm a senior lecturer at the University of Limerick in chemistry. And my research mostly focuses on actually drug delivery of therapeutic molecules. And what I tend to look at is molecules like peptides and proteins and biological molecules that are difficult to formulate into a medicine. And that's one of the challenges when you've identified the molecule that will have the right biological activity. It's how do you package it up so that it can be active when you take it as your medicine. So that's kind of my angle.
Very good. Thanks, Sarah. Abina?
I'm Abina Crean. I'm a professor of pharmaceutics in the School of Pharmacy in UCC, and I'm also a pharmacist. And a bit like Sarah, I'm really interested in how we can deliver these molecules into the body, and how we can, once these molecules are identified, be them vaccines or therapeutics, then how can we manufacture them efficiently? Because they all have certain properties, they all have certain characteristics that we need to address. So how we can manufacture them, and then how we can deliver them into the body, into converting these molecules into medicines that we take, like injections, patches, tablets. That's my area of interest.
Very good. Thanks, Abina. Peter?
Welcome, everyone. So I'm a lecturer at University College Cork, and I'm a biochemist by training. And similar to previous speakers, I actually focus also a lot of delivery of complex molecules, in particular focusing on nucleic acids that are also being parts of the vaccines, like the messenger RNA, for example, and trying to deliver these molecules into specific.
Oh, it's gone.
You've gone back on mute, Peter. Mysteriously, something. Yeah. There we go. You're back again.
Yeah, so trying to deliver this molecule in specific places in the body to enable treatment for diseases that currently don't have effective drugs available.
Very good, great stuff. Anne?
Hi everyone, my name is Anne Moore. I'm a senior lecturer in biochemistry in UCC. I used to work with Abina in the School of Pharmacy. My background is completely vaccines. How can you make vaccines for, I've worked on every disease from HIV to Ebola to flu and malaria and other diseases as well. And it's all about understanding how you can make a vaccine, understand the immune response, and then translate it to humans. And as part of that, I used to work for many years with the what are now called the Oxford Group, who have developed the AstraZeneca vaccine. So have a good insight into how to make vaccines and test them. So I'll leave it there.
Great stuff. Thanks, Anne. So I hope everybody online has got a feel for what we're all doing. And you've got great expertise here, obviously, because Anne's coming at it from the vaccine point of view. And the connection to Oxford is very strong, obviously, there. And then Peter is into RNA delivery, let's face it, which is a big area for these new vaccines that have been developed. And then Abina and Sarah are more into therapeutics as well, and drug delivery, I suppose, in many ways. I think we'll start maybe by each of us giving like a 5-minute account, I guess, of where we think it's at for the moment with COVID, at least as specifically as we can, and what the questions that we see as being important are from this new medicines point of view. Because remember, this session is about vaccines and new medicines. And also, any questions, we'd love you to send them in. Sarah Hayes is going to coordinate that. So the more questions we get, the better. We have a fair few to go through, but some of them group into sort of similar categories. But please don't hesitate to ask us any questions at all about vaccines or therapeutics. They're the two main things we're going to talk about. So I might kick off from my point of view. So I've worked on septic shock for years, and that is an overwhelming systemic response to infection. And it happens with bacteria, it happens with viruses. And I'd worked on gram-negative sepsis, for instance. Also worked on certain virally induced septic responses as well with certain viruses. So when COVID broke, my lab was kind of well placed to switch some of the projects into the more therapeutic aspects to do with the inflammatory events. And in essence, there's about 1,000, I think, different medicines, shall we say, or therapeutics in development for COVID-19. Because even though the vaccines are one way out, of course, we all agree that's the great triumph that will happen. There'll still be a need for therapies of various kinds. And secondly, even outside COVID, the big hope will be if we discover new therapeutic approaches for COVID-19, they may well apply to other inflammatory situations and other infectious inflammatory diseases as well. Because remember, sepsis remains a major clinical problem. Even though we've known about sepsis for a long time, there's still a big need for new insights and new approaches. And specifically in my lab, we are working on a new anti-inflammatory molecule. It's called itaconate. It's made naturally by the body as an off break for the inflammatory process. We found this back in 2017, I guess. And now we're trying to figure out, can we turn this natural anti-inflammatory into a new therapeutic? And amazingly, 2 labs are beating us with COVID. They've already seen itaconate in COVID. It goes down with severity, which is interesting. And remember, it's an off switch. So if it goes down, that means you might get more severe disease. And that was discovered through just what's called a metabolomic analysis of COVID-19 patients. And then secondly, another lab has found if you give itaconate in models of COVID, it can protect against severe disease in the hamster model. And we've now repeated that ourselves. And we're making derivatives in various ways. And that's kind of a major focus for my lab. The other thing to say is, very briefly, this disease is a coagulopathy, which means you get massive microvascular coagulation in tissues. And again, we're finding new ways to block coagulation as a possible therapeutic approach. And again, things like itaconate, a thing called the NLRP3 inflammasome. My lab works on that. They're tied into coagulation as well. So again, we're using kind of new ideas, I suppose, to go after COVID, which could be useful therapeutically if people get the disease, or down the line in terms of other diseases, we may see applicability there. So there's a quick snapshot from my point of view. Sarah, do you want to go next?
Okay, yeah, thank you. Thanks, Luke. So when Sarah Hayes approached us about this panel, it was making medicines in a global pandemic. And I could see from some of the questions that we've already seen come in is that people kind of keep asking, well, are these medicines being rushed? Are they coming really quickly? And what I'd like to get across, and what I think has been so important during the whole making these vaccines during the pandemic, is that we're building on decades of drug delivery research that's already been out there. So it's not that they're being developed very rapidly. they're getting to the market rapidly, but the development has been in place for decades already. Peter will talk later, I'm sure, about some of his work he's done on using lipids for RNA delivery. You know, 20, 30 years ago, the lab I was in was working on screening thousands of lipids and changing their structure so that they could deliver these molecules. So, you know, Luke identifies the molecules that we need to get into the body that will have the effect we need. But to get them into the body and to make them into the medicine, you have to basically, like we said, package them up. And so part of the reason that we've managed to get the medicines or the vaccines developed so quickly is that we're building on drug delivery technologies that have been in development for RNA, also for peptides and proteins. And what's really exciting from our perspective, or from my perspective as a researcher in drug delivery, is that we're actually now getting the chance to really implement these into a vaccine that's getting out to patients, which would have been a lot slower if the need wasn't so great. But the need is great, which is expediting things. And I think that's really important. I think people have really shared expertise and techniques on how to develop the drug delivery aspect of it. And, you know, what we've developed so far are vaccines that have to be injected, you know, by a healthcare professional, which is fine in the Western world. We're in a developed country, but we still need lots more research where we can get these vaccines where keeping them cold isn't such an issue, where we can develop a delivery system that stabilizes them so that it's easier for people in less developed countries where they don't have such access to healthcare professionals or to refrigeration and things like that. So while we have vaccines that are available now, there's still a lot more to be done to develop medicines that can travel easier and be more accessible to everybody. So just to make that point.
Great, thanks, Sarah. One phrase I heard today, just now, I've come off my COVID-19 centre call, for instance. Ed Lavelle was saying it took 100 people 10 years to make those liposomes, didn't it? It was a huge amount of effort to get to the point where Moderna and Pfizer could use those liposome delivery systems. People don't realize how much time and effort it's taken, do they? Though?
Yep, sorry, just a delay on muting myself. Yeah, and like, you know, there's a lot of thought goes into that. They look at, say, for example, delivering a protein. You look at the structure of the protein and you design the lipid so that it can, first of all, get a lot of the protein or the RNA in there and then safely deliver it. So there's a lot of design strategy in there that's really has been being developed for years.
Yeah. Great stuff. Thanks very much, Sarah. Abina?
I suppose the area that really interests me is this storage challenge. And I think we're all well aware of that. We're now, for years, we're talking about removing cold chain. And because vaccines in the developed world are refrigerated, it's not such a challenge for us in the developed world, but in the developing world, even refrigeration is a huge challenge. And that last hour or the last kilometer. So for a long time, we've been working on, again, these protein molecules and working with Anne on the vaccine side on developing solutions that eliminate the need. So the idea of freeze drying these molecules. And I think around a third of the protein therapeutics on the market are currently freeze dried. And it allows us to stabilize them, and it allows us even to store them for periods of time outside the fridge. And we're really now starting to see the benefits to healthcare of that. I think it was kind of the poor cousin to, like, everybody wants to deliver find the cure to cancer, find the cure to COVID, but we're now seeing all the small little hurdles that have to be overcome. And in our lab, the freeze drying is one solution. And even freeze drying, it's a well-established technique, and it's a slow technique though, and we're now seeing the need to get, I think Pfizer want to have 1.3 billion doses. That's phenomenal. I think Pfizer last year produced 200 million doses of vaccine. And now they're going to, on one vaccine, they're going to have 1.3 billion. So able to do things faster, more efficiently is really needed. So I think this healthcare challenge has really pushed us to take to implement faster techniques that I suppose the industry is quite conservative and it's held back on it. But now I think those solutions are in research, and now we're seeing them translate into products on the market. So that's, I suppose, the real area I'm quite passionate about. And the other area is the injectables. And Anna and myself, as Anna alluded to, we've worked together before, and we work in this area of microneedles, you know, trying to have an easier way of delivering instead of an injection where you need a trained healthcare professional. So I'm a pharmacist, and hopefully the pharmacists will be delivering this as well as the GPs, but that still is a limitation. So I think the vision for the future is if you could ship these medicines to people and they could administer themselves like a Band-Aid or a patch. Now, that's not going to happen in the next year or two, but that's where the research needs to go. So when we have a pandemic, we can ship it out to people, and we can have devices, maybe not a microneedle, but some kind of patch that people could use, and or maybe even oral vaccines. But because of the stability of these molecules, that's a substantial challenge. Yeah, so
is it true, Abina, that Pfizer are now freeze drying that vaccine? Is that I read that
I
yesterday?
imagine they are, and people were saying, well, why are they storing at -70 and the Moderna's at -20? And they're similar type compounds, but I would imagine it's, and I've spoken to people, maybe not in that company, but other companies, and the challenge is to get into man fast. So they probably took, this is speculation as a scientist, but I'm speculating for other products I've seen it happen, where they'll say, well, we want to get it into the market. So the best chance is to store it at a very low temperature, because what happens is, you know, they need to have, for the regulators, so much stability data to get approved. So they need, you know, they obviously focus their effort and they have good stability data at -70. They probably have a second, stability study at a lower temperature, and there will be, I would definitely say they're freeze drying. They're looking at freeze drying it, and I would say there's a high chance that will work for that product.
Is that just not a step in the reconstitution in the vine? Is it? In other words, if you freeze dry it, it's just one more step. Is that the way to think of it?
Yeah, it's reconstitution. I think the Russian vaccine is freeze dried. I think
right.
The Russian. It's not a challenge. The freeze drying, especially for this product, it's not going to be a re-challenge to reconstitute it, because you've got so little RNA, and so I suppose that you can encase it in lots of sugars that dissolve fast.
Right.
But they will have to reconstitute it, and they will have to check that the lipid siRNA structure maintains within that, you know.
Yeah.
So I suppose, actually, now I'm thinking through it, that's a key step. It's not that it'll take time to redissolve it. It's that arrangement of the siRNA and then the lipids stays intact long enough so that it's still effective. So they'll have to do studies to look at that. But I would imagine freeze drying is going to, it will be a process that's involved.
One of the quick one, and I'll dominate the slightly, but I got a great email yesterday about a 10-year-old, wait to hear this, who said about microneedles, right? And they said, could you make a lollipop with tiny microneedles and a kid would get the vaccine that way? I thought, what a great idea. Could you put microneedles into a lollipop?
Yeah,
might be possible, but often these ideas come from the most, you know, our youngest friends often are the most innovative, aren't they?
Yeah, I think it is. Now, not to put a dampener on that 5-year-old enthusiasm, the biggest challenge, one of the big challenges with the microneedles is, I suppose, when you have an injection, you have a volume of liquid in a syringe and you know that's gone in.
Yeah.
You know, with the microneedles, you don't know how many went in, you know, especially if you're giving a kid a lollipop.
Yeah.
I have 2 kids and they eat lollipops lots of ways. They bite them, chew them, suck them. So
right.
There'll be a huge variability, but there could be things like a little patch that you just stick into the side, and that, you know, that the parent applies a certain amount of pressure every much. So, or even a sherbet flavored.
Yeah, you never know. Yeah, exactly. Very good. Thanks, Abina. Peter.
So I suppose I think my lab and I in general, I'm really captivated by the potential that RNA molecules bring as therapeutics. And as Sarah mentioned, the delivery obviously is a central part of that. But I'm really excited about the fact that molecules that I've been working with since my PhD are now actually almost after 2 decades of development actually turning into the real product that comes with siRNA being a actual drug right now, as well as messenger RNA being a first approved vaccine. But there's so much more potential that can be harnessed from that molecules we're talking about cancer vaccines, we're talking about gene therapies, protein replacement therapies, all sorts of things that can be done. And obviously, delivery here is one of the central pieces to the puzzles that needs to be solved. All the issues that Abina talked about, how do we going to store those drugs, produce them at mass scale? So there's lots of challenges, but what really keeps me going and working about RNA molecules or our own RNA molecules, it's actually this potential that we can really bring a new class of drugs on the market and help to cure diseases that are currently not really possible to cure with other ways.
Quick question, Peter, then while we're at it. So obviously the idea was probably first configured, 20 years ago or more, right? That RNA could be a potential drug. What, just for the audience, what were the two key barriers or challenges that people confronted from that moment on? And then they've obviously been solved to some extent. But what do you think were the key issues about using RNA as a drug or a vaccine or whatever?
Yeah, so I think the key issues for both smaller RNA and messenger RNA was one, delivery. So these drugs have very low bioavailability, which means that they have no means on their own to enter the cells, or very little. Therefore, they need help. And figuring out how to design this kind of package them into some kind of a delivery system and make this delivery system to go to a tissue of interest or a place of cell of interest was kind of critical piece to the puzzle to solve. In case of messenger RNA, the other key component also for smaller RNAs like siRNA was how to stabilize those molecules, because RNA inherently is a very fragile molecule, much more than, for example, DNA. That's why it was much harder to work with and kind of neglected in drug development for many years. But there has been key research done on modifications, chemical modifications to nucleosides to make those molecules much more stable. And these were actually, I would say, the two main features that enable application of those molecules. The second, the modification part also solved another problem with application RNA as a drug, which is immune response that actually those RNA molecules cause. So viruses, our body is equipped with the mechanism to deal with viral infection, and viruses are often made of RNA as well. So we are able to react to the RNA that is coming to our cells, and those delivery systems mimic kind of a virus. So modifications were key piece to the puzzle, also not only to solve stability, but also hide these molecules from being recognized by our own cells as a kind of foreign or viral DNA and prevent them from reacting.
Right. Were they separate modifications, Peter? They were they? The stability one was different to the one that would allow it not to be so immune stimulating, shall we say? They were quite different, were they, I presume?
So there is an ample of modification that can be applied. Usually those modifications are applied to uridine, but the siRNA modifications are more somewhat synthetic, chemically based. But for messenger RNA, these modifications had to be kind of natural. So people look at modified nucleosides that exist in nature, because the mechanism how both RNA molecule works is completely different. So in case of mRNA, where you have to make a protein out of it, you need to make sure that your ribosome, so that the kind of machinery that reads the message is not getting stuck. And if we would introduce some kind of foreign modification, it would probably obstruct this ribosome. So therefore, the origin of modifications was different for this RNA molecule.
Right. Again, I think it really illustrates anybody listening in who's a young scientist, how much science has gone into this. I mean, not only do you have to worry about the structure of the RNA molecule, you got to worry about immunostimulatory properties, and then with the liposomal making the fatty bag, shall we call it, massive science went behind altering the lipids and the fats. And in the leaving cert, you learn about the lipid bilayer. I hope still anybody listening who's in the leaving cert, but that lipid bilayer is very complicated. To make that into a delivery mechanism took a huge amount of effort in science, didn't it really? So I think it's a great example, isn't it, of many people, of course, over the course of 20 years, all giving a little piece to the puzzle to eventually crack it, I guess. Now, we know a couple of people who've made a big difference, of course, but overall, it took a huge amount of effort, didn't it, really? Very good. Anne, we might move on because we could be here all day, couldn't we? But Anne, you're next.
So I guess my journey is kind of similar to a vaccine. I started off actually with doing my PhD with Kingston Mills long time ago. And it was all about HIV vaccines and how you add these molecules or materials called adjuvants that can modulate the immune response to a type of response that's most appropriate for that particular infection, be it a virus or a parasite. And I was also interested in the route that you give it, whether it's, you know, into the muscle, into the skin, into the mouth, etc. And I suppose over time I got more interested then in well, it's all okay, you know, at a preclinical level, you know, in the lab, kind of coming up with these great ideas, but how do you triage out what actually will make it in humans and be a good result clinically? And I suppose then the last, 20 years, especially when I was in Oxford, you know, we really focused on pandemic preparedness. We all realized, the whole community of vaccine experts in the field of vaccines knew that there was going to be a pandemic. And we've seen some scares even in this century with Ebola and SARS and Zika. And even the H1N1 that happened in 2009. And we learned a lot from that as a vaccine community, what we needed to do better, what we needed to do faster, what worked, what didn't work. And I think even though they were near misses, and for a lot of people, they weren't a near miss at all. They suffered greatly from those diseases. You know, this time, as Sarah was mentioning, it's taken 20 years of hard work to get to 6-month overnight success. And really, like, my interest in that is understanding if we can develop what we call a vaccine platform, a type of vaccine. So people are more familiar now with RNA or with adenovirus, and they'll become more familiar with subunit. Can we use that in the future for, well, we know this induces this type of immune response, therefore it should work for Ebola, SARS, coronavirus, etc. And as on top of that as well, what we found is that using a different route can really solve some of the issues as well. So Abina and I have worked a lot on developing vaccines into these little skin patches, these microneedle patches. And we found some really interesting results in that, how you can get broader immune coverage if you go into the skin compared to if you give it into the muscle. And I suppose, so that again is all part of developing vaccine platforms that give you the right immune response and allow you to stabilize the vaccine out of cold chain, as Abina mentioned, and also allows you to deploy it really easily. Like, one of the great successes of polio vaccine or poliovirus almost eradication in the world is because it's so easy to give an oral polio vaccine. It's just 2 drops into a child's mouth. You don't need that much training to do it. So there is a real need to develop these kind of alternative vaccine formats, so solid dose or, you know, drop formats. And so when I moved to pharmacy, I'm very influenced by my environment. So I started working on those kind of new ways of approaching things. And I suppose then, you know, the other thing that's great, and you can develop a fantastic vaccine, but if people don't want it, as Mike Ryan at the WHO said, a vaccine is no good in a vial. People have to want the vaccine as well. And again, with colleagues in the School of Pharmacy, we started working on vaccine confidence in Ireland, and that's a whole other kind of way of approaching things and understanding. You know, scientists have spent a lot of time looking at developing vaccines that work, but if either people don't want them or, you know, ultimately the market isn't there. You know, when I was working in the States way back in the late 90s, we developed an Ebola vaccine, which worked perfectly well, but there was no need to develop it because there wasn't an Ebola virus outbreak. So it's all these kind of influences that determines whether a vaccine is developed and used or not. And I think, you know, on some of the backs of some of the things that we have done in the past has allowed us to develop these vaccines really quickly in 2020. I have to keep reminding myself that we're not in 2020 anymore. And really trying to develop that. So at the moment, when it comes to COVID, we've worked with, I have a collaboration with an American company making an oral tablet vaccine, and we could get really obsessed on the kind of structure and the amino acid sequence of these proteins that are used as antigens, and understanding what is the best protein from SARS-CoV-2 to put in these vaccines. And it's been an interesting ride, to say the least. So in the interest of time, I'll stop there.
Very good, thanks Abina. I mean, what you're saying strikes a chord with all of us, of course, you know, and about my own journey with therapeutics. I mean, we got our molecule in 2016. We made 10,000 versions of that to get the right one eventually, because of all the different, what they call PK/PD. So I think, as Sarah said, it's all very well to have a new approach, but there's so many extra things you need to bring that into a human and develop it further, I guess. So, and the vaccine business is the same in a sense. Why don't we move on with some of the questions, shall we? We've touched on a couple of things, but now the danger here is it's all vaccines, and so you better get ready. I think 2/3 of them are about vaccine, which is okay. But But a big one, and you've touched on it yourself, and I keep getting this, I'm sure we all do, the speed question. And I know that in America, I heard yesterday, lots of the Pfizer vaccine is still on the shelf because of hesitancy. And it turns out when they ask them why, it's because this was developed too quickly. So what's your response to that worry that people have that these vaccines were developed far too quickly, questioning the safety issue really, you know?
Well, I think when it comes to speed, Abina is probably the person to talk about the manufacturing quality behind it. I guess from a vaccine clinical trial perspective, like up to now, every time you do a vaccine clinical trial, it's very sequential. You only have enough funding to do that trial. You get the results, you go back to the funder or another funder, and that's what takes so much time. And it's one of the key things, hopefully, that will come out of all of this, that science needs to be funded pretty well so that we can make these often incremental increases, but that there is the funding there, that you're not spending the next 3 years just trying to make the next step. And that whole issue of having funding has been completely wiped out, which is great for COVID. So that's why we've managed to go faster and do things in parallel, because the funding is there. And I think Abina is a really good person to kind of describe the quality that goes into developing pharmaceutical products.
I think as well, and Sarah Gilbert said that just there was no need to write grants anymore, which takes a huge amount of effort. You know, in other words, the bureaucracy of trying to get money in and do all the Gantt charts or whatever the hell it is was wiped away from this, which was a big effect, wasn't it? Because that allowed things to go much faster as well.
Yeah.
Abina, do you want to give us your view on the manufacturing or the other aspects?
I suppose the manufacturing, and as well as the manufacturing, the regulatory process that it goes through and the inspections, like I suppose we're lucky to be part of the European Medicines, or a member of the European Medicines Agency, and they're really well, they have a very good reputation. I would say as a regulatory body, they don't bend to political pressure, and we saw that, you know, they waited till they'd done all their work. So there's always, there was no shortcuts taken with this, and all the trials were done. The data was looked at, you know, there could have been a pressure, let's get it out there faster. You know, we have waited for this, you know, the trials are done. They did spend time looking through it. So I would have great faith in the European Medicines Agency for the work they've done, and our national agency, the HPRA. And then on the manufacturing side, you know, the pharmaceutical industry is so highly regulated, and anyone who's got people who are relatives or anyone who works in it, I know down in Cork, a huge number of people work at the pharmaceutical sector. As I say, you can't scratch your nose without filling out paperwork. You know, everything is documented, the quality of the components that go in each step. There's a huge validation, huge testing. So I would have great faith in the quality, you know, and by quality I mean the reproducibility of each batch of the product, and the safety from the development through the assessment of it. So we're in a very highly regulated sector, and that sometimes frustrates people because they want medicines to go, why can't we, that medicine's discovered, why can't we have it now? And there's always that play between, you know, the good, for the good of, you know, there's certain risks, but I think for this vaccine, it hasn't been rushed. It's happened faster, but that doesn't mean there's any shortcuts being taken along the way.
And it's true about it that they go to the factory, don't they, and actually test the batch and everything. That people don't realize how diligent that is, right? It's not a case of ticking a box here. They actually go and take the batch and really analyze. The EMA analyzes it themselves, don't they?
Yeah. Well, what they do is they have a pre-approval inspection of the site, and not so like they will analyze it themselves and they can take a batch, but because you can always have actually with making pharmaceuticals, the testing is, you know, the quality isn't built in testing. Testing only tells you what you have, and you're only testing for your known risks. So really a lot of where the quality lies in is the controls of the raw materials, making sure that there's no contamination of the raw materials, making sure the equipment's safe, the packaging line, the air quality. And what they're doing when they go and do those inspections is checking the records on that, and really checking the quality systems of that factory, because you can go in and, you know, as I say, the Queen never smells anything but fresh paint. You know, you can look good in a day, but what they're doing is going back historically I've heard of an Irish inspector who went around with a dental mirror and he went in under machines looking for dust. You know, these are rigorous inspections.
yeah.
The quality of is very good. And actually Ireland has an excellent reputation. One of the reasons we're so successful is our pharmaceutical quality is really good, and we've had very few citations. Actually, I don't know if we've had any major citations for poor quality.
I think it's fair to say, Abina, would you agree, in terms of regulation, the pharma sector has the highest of all standards in a way, for obvious reasons, and people should be reassured by that, shouldn't they?
Yeah.
The other one is the airline industry, apparently, because you can't have a plane that isn't able to fly properly, you know.
Yeah.
Usually with medicines, it's extremely high bar, isn't it, that you got to pass to get any kind of a, you know, validation or regulatory approval.
Yeah. And as well as that, before the product can be released, they have to have huge stability data, and they also have to have had this validation. They have to have all their process validated. Yeah. So I think it's as high as the airline. And
I know, but it's so reassuring, and people don't realize this, do they? And that's why it's good. You can explicitly explain it on this call we're having, because people don't realize how stringent it is.
Yes, the other thing as well that has been really reassuring for the vast majority of these trials is the actual rigor of the clinical trials, how well they've been set up. And, you know, the findings are based on a high level of confidence because the clinical trials were set up so rigorously with a very, very high number of individuals, and all of the results are very transparent. You can dig into all of the results of the trials that have been published and see exactly what happened. So instead of looking at you know, the last tweet or the last, you know, whatever social media piece that something happened, you can actually go and see what happened and see, you know, efficacy isn't based on 10 individuals. It's based on hundreds that actually got infection, of thousands that were actually immunized. So there's a huge database to give us confidence of what these producers of vaccines are saying that they do. So that is really good.
Exactly. Yeah, but hopefully we reassured the person who said they would take the vaccine out of a dirty sock if they could be reassured. So maybe they'll now be prepared to do that. But I think that's extremely useful to everybody, I guess, who was worrying about this question. We might move on. And another question we have, maybe Peter might have a go at this one, the difference between the RNA approach and a more traditional approach. Could you give an answer to that one? Because obviously, again, that's coming up a lot, because we now know there's at least 3 RNA vaccines, one because I think CureVac is RNA as well, isn't it? But the others are more traditional, aren't they? What do you think about the difference between those approaches?
So actually, that's a little bit trick question, because there has been over, I think, 180 vaccine candidates at different technologies that have been entering clinical trials. So obviously, the second, I would say the next one in line is AstraZeneca's vaccine, an Oxford vaccine, which let's say it uses more traditional technology, but has not been actually a vaccine approved based on that particular technology just for human testing. So it has been, I think, for rabies, but so in animals, but I'm not sure if there has been one for humans. So basically the difference maybe between adenoviral, so AstraZeneca's vaccines and mRNA vaccines is that AstraZeneca vaccine uses DNA molecule to train the immune system instead of messenger RNA molecule. And instead of being packaged in synthetic lipid nanoparticle that we talked about, is packaged into a viral vector. So in this particular case, it's adenovirus, a somewhat common cold virus. And this particular one was taken from chimpanzees. So it doesn't cause the disease in humans, but causing the disease in chimpanzees. So it should be safer. It's also not able to replicate. So there has been many safety improvements into that vector. I think in terms of production, the difference is that mRNA is newer technology. So the scale up of the process is kind of manufacturing is still have a lot of, things to be, let's say, maybe developed or require more development, whereas the viral vectors like adenoviral vectors have been used already in the field of gene therapy. So manufacturing is kind of more streamlined. Therefore, these vaccines have the potential to be cheaper compared to at least mRNA vaccines right now. And I guess that's probably kind of that would be, there is also different in terms of storage, since the DNA molecule is more stable. Those vaccines could be stored at 228, so like refrigerated temperatures, whereas mRNA, at least for now, with the current formulation, required more stringent kind of cold chain for transportation and storage. But I think then And more can probably also add some more to that because she's been working with Oxford vaccine.
Another quick one, Peter, actually, that's I think I get asked as well, is why is the RNA one so much more expensive? I know you touched on it there, but we're looking at like maybe 10 times the price, aren't we, compared to, say, the Moderna vaccine, if you compare the AstraZeneca one?
The estimates are about $2 to $5 for price of Oxford and $20 to $30. So I would say it's just, at the moment, it's more expensive, probably because indeed there's kind of gaps in manufacturing and that it's been relatively new technology. So what I can say based on my own experience with mRNA research is that since I've been a postdoc, the price of making RNA actually went down. So it's kind of somewhat like with sequencing. It will require time for the process and reagents and things to be optimized. And I guess the other component is that the lipid nanoparticle is also a second part of the vaccine. So it also adds to the cost of manufacturing. So yeah, it's just more complex. Yeah. Somewhat technology at the moment. That's why.
Sarah, are the lipids expensive? To make that liposome, is that a costly thing to do anyway? Because there's a number of lipids, isn't there? I imagine in the liposome, it's not just one. So whatever that composition is, I wonder if that expensive.
It does depend on what lipids you're using, but yes, I would say they are expensive.
Yeah, and then the supply chain issue I read as well was tricky. They couldn't get some of the component parts quickly enough, could they?
Exactly. Yeah.
So that's the mix, I guess. We might go back to Anne now for a few immunology questions because there's a fair few here, Anne. Are you ready? And I know you know the answers, so but why do we need a booster? Give us the story behind the 2-dose thing.
Luke, you're the real immunologist
here. I'll let you answer that.
Well, you need a booster. I mean, it is very much like being in school. The immune system is no different to all of us when we're in school learning lessons. You see the material for the first time. You see something that's, in this case for the immune system, it's a protein antigen. And you have to figure out how to respond to it. Your immune system figures out how it will respond. But to really hammer the lesson home, you have a second, you see that material again, and that allows you to develop a faster and stronger and more efficacious immune response. And that's all, we could spend hours talking about maturation of the immune response, but we won't. And it is really about learning the lesson as best you can. And all vaccines, I mean, every childhood vaccine, and bar maybe 1 or 2, are always given in a 2 or even 3-dose regime. So it's not unusual at all to have more than one. And to some extent, it's a little bit pragmatic sometimes, like some vaccines, I won't say which one that's used in adolescents, you might actually be able to get away with a single shot of that, but overall, it's a kind of pragmatic as well. It's not everybody's going to respond exactly the same way. So some people aren't going to respond as well. So give them a second immunization to make sure that they'll catch up with everybody else. So it's pragmatism, but it's also for your immune response to develop the fastest, strongest, most efficacious response.
Yeah, I like your analogy of the school. I think I had an analogy myself yesterday, which I realized was wrong. I said the first dose gets the players on the field, and the second dose is the whistle to get them playing. That's not quite it, is it? It's more the players are on the field, and then the manager starts shouting at them. Maybe that's a better analogy.
Exactly.
The second dose is the shouting from the sideline.
And it's going to keep practicing your keepy uppies, and you'll be like Maradona.
That kind of thing, exactly. Yeah. Another quick one as well, and about the a big question is how long will it last? Have you got a view on that? We don't know, do we? I suppose the answer.
No, but a really important question that will answer that question is, what is the minimum threshold of immunity that you need to maintain protection? So, like for some vaccines that we work on, we know that we will need a huge amount of antibodies to maintain protection. And therefore, if you have a vaccine that you've just a very quick peak and then it goes down, you lose that protection pretty quickly. But if you only need a very, very small amount of protection, a very small amount of antibody response to keep you protected, it's likely that these vaccines will last much longer. And there's a tiny bit of data coming out of some monkey studies saying that actually they can be protected with a very small amount of antibodies. How that translates to humans, but it's good to see that.
Yeah. Would you call it, Anne? Do you think it'll be every year or every 2 or 3 years, or what do you think?
Yeah,
not possible to answer probably, but I think with SARS and MERS, they got 2 to 3 year T-cell responses after the first shot, didn't they?
After
that gives us a bit of hope, doesn't it? I suppose it might be every so often. It won't be every 6 months or a year, probably beyond that maybe. But,
yeah, and I mean the infection, like the studies that have been done after infection show that you do get good memory if you get a bad dose of virus, which, you know, you don't want a bad dose of virus just to get that, which is why you get a vaccine. So hopefully, I think the vaccines should give us a nice memory immune response. And we'll see in the first few months of this year how it's looking, but hopefully it won't be a seasonal one. But again, that comes back to the factor of how much this virus changes and mutates and drifts over time. Hopefully it doesn't, but.
Well, we got to answer that question next then, don't we, really? The variants that are emerging. What do you make of all that?
Yeah, I mean, this is why we need to vaccinate in a timely manner. We've got to get people protected as soon as we can. It is, you know, otherwise, I mean, that variant in the UK seems to have appeared in a cancer patient who was under immune suppression. So their antibody responses were absent if they're at all, or sorry, even if at low amounts. So the virus has more time to live in a person and has more chance to mutate in this kind of very average kind of immune response. So it has more chance to see what the immune response is doing and then avoid it. And similar to flu, start emerging out of that then. So it's you know, we need protection as soon as we can. We need to build that barrier across the community as quickly as we can in a timely manner.
Yeah, yeah. And lastly on this point, I suppose, do you think, you know, the debate at the moment is, can you get away with the one shot and delay the second shot, or maybe even avoid the second shot? What's your opinion on that? I presume you'd be like me and say, no, stick to the protocol.
Absolutely. I mean, you know, we did, well, not me, but, you know, evidence-based, rigorous clinical trials were done, and the results are clear from those. And we have confidence from all of that data that if you give 2 doses 3 or 4 weeks apart, you really significantly increase your chances of protection. So we should do that. With the Oxford AstraZeneca trial, I worked on that, and I still work on adenovirus, and I've worked in there for years. And there, I mean, we have papers on the interval between the first and second immunization. And for some of those, you can give it after the second immunization after a long duration. But you know, we always develop those vaccines as not for a pandemic. So we'll see what happens.
Yeah, I guess the pressure is from the variant in a way, isn't it? And the supply issue being threatened, or not as confident as people feel. But I think most of us agree that you should follow the protocol that was established in the trial. It's the safest thing to do. We might move off vaccines for a minute. We'll probably come back to it, mind you. And Sarah, there's a good one here that was sort of flagged up as well. Why can't we make a medicine to combat the virus instead of a vaccine?
Why can't we make a medicine to combat the virus
instead of a vaccine?
Oh.
Or in other words, can we not get rid of the virus without a vaccine? I suppose the question.
I suppose you have to think about, where does the virus go when it gets into your body?
Well, I guess that question might partly be from HIV and hepatitis C. We didn't get a vaccine, did we? And yet there was a good antiviral thing. So what do you make of the whole non-vaccine strategies that are out there? Or is there any hope in those, do you think?
Again, Luke, you're probably better equipped to answer this because you work with antivirals. But I mean, my thought is that, you know, antivirals usually don't manage to completely kill off the virus. Would that be a fair comment?
Yeah.
Whereas your vaccine should. And as long as there's some virus left, it's more challenging to combat. I mean, you're basically, you know, you heard people talking about how after COVID the virus remains. I remember hearing people say, "Oh, it's in different parts of your body," you know, and it's dormant. And that is probably true, but the vaccine would be able to combat that.
That's part of it. Yeah, I mean, the other big part is those other ones I mentioned are chronic viral diseases, HIV and Hep C, remember? Yeah. And there's one view that it's easier to treat a chronic disease with an antiviral. These acute ones
keeps the symptoms at bay
because they take off so quickly and you can't really combat them then. I mean, there is some evidence that remdesivir, which is the one that's been developed first, does work a bit, mind you, but it's not great, you know? Same with Tamiflu, you know, the anti-flu one, that never really worked. Did it, Anne, do you think? They were flogged as possible therapeutics, but didn't really work, I suppose. And then I think from the anti-inflammatory point of view, clearly we need new therapies anyway, you know, because there will be a subset of people who will get very sick with these viruses, or this virus, and specifically COVID-19, of course, and the vaccine won't be the only answer potentially here.
Now, would you
Although
still
there's a-
want to, sorry, Luke, you still want to be able to treat the people that have the virus?
Exactly. Yeah. And I mean, it's a broader question for us all that I'm getting a lot now is like, will the vaccine be the actual answer? Because, and can we imagine if we get, say, 6 months from now, are we now out of this nightmare? And of course, there's two schools of thought. One says that as soon as the death rate drops, and it's not as soon as it'll take a while, but the death rate will come down dramatically with the vaccine program. Of course, it will. The level of illness will go down as well. Then it becomes like a more normal disease, which means governments can't be justified in their strategy anymore to lock down the place or have restrictions in a way. The counter argument to that would be, by the way, people will still get sick. There won't be full vaccination for a long time. Young people might develop long COVID, which is a worry. You know, those sorts of concerns means that the vaccine is not necessarily the magic bullet that we're looking for. Is it, in a sense? I mean, does anybody else want to try to answer that one? Because
Guys, I might just, oh, sorry for interrupting. I might just jump in a moment. You guys are amazing. You are so fascinating. I think we need like 4 or 5 hours of this, but we have only got 8 to 10 minutes left, and there's a load of questions coming in here that I would love to just jump through and do a little bit of rapid fire with you. But I think some of what you were just about to launch into or discuss, Luke, will come up.
Yeah.
Would it be okay if we just
fire away?
put you with some questions? So this is a great one. This is from Christian, who's 11, and they want to ask you specifically, Luke, can they defrost the vaccine before they inject it into people? So I think we're talking about the Pfizer, BioNTech or Moderna vaccines that are being kept at minus 70 degrees Celsius. So do we warm those up to body temperature before we inject them into people?
Oh, we do indeed. You can't be injecting frozen stuff unless it's an ice pop. But no, that's a good question, obviously. Now, you got to thaw it out. Yeah, I wouldn't say it has to be body temperature. Does it though? Abina, it can be room temperature or lower.
It's usually room temperature,
Yeah,
and
room
they
temperature,
thaw
yeah.
out gradually. And it's a good question, because I not this vaccine, but I heard of another product, and from a patient that they were defrosting it in their microwave. Not recommended.
No, that could destroy it, right?
Yeah, it would destroy it. So these have to be defrosted, like just left at room temperature and defrosted gradually. No such thing as no rushing this process. Has to be left to defrost gradually. And once they're defrosted, I think they can be stored at room temperature for a number of hours then at that stage.
There's even guidelines on the Pfizer vaccine. You take it out of the really cold freezer. It takes about 3 hours to fully thaw the box, and now it's ready to be used. And then it's reached room temperature after 3 hours, I guess. So that's the recommendation, obviously.
And you can't refreeze it. Once it's thawed, that's it.
Yeah. Okay. And that's the issue then with transport and actually getting it out to nursing homes, whatever. That batch has to be used once it's been thawed out. Okay, great. We have another question from Sarah and Emily, 13, and they know that some vaccines have allergen information because of how they're developed and the fluid that the vaccine might be in. What allergies might prevent you from getting any of the current approved COVID vaccines? Are you aware of any that might prevent you from getting any of them?
I can have a go as well. I'm sure Anne can as well. I mean, there's a thing called polyethylene glycol, which is in the Pfizer vaccine. And they reckon that was the thing that provoked the allergy. And there was a very small number of people. I think it's 10 people so far have had that allergy to this. The polyethylene glycol is used across many different things, not just for vaccines. So that was the reason for that. Famously, the flu vaccines are made in eggs, and people have an egg allergy. So that was the recommendation not to take it if you're allergic to eggs. But with this one, there isn't any other warnings about allergens or possible allergic reactions, which is good. You know, we wouldn't worry about that one at the moment, actually.
And those allergies, those few instances of severe allergies were in people who already had severe allergies and carried EpiPens. I don't think there was any new severe allergies of people suddenly presenting with allergy after being vaccinated, from what I know. So if you have a severe allergy and you're going to be vaccinated, just let the person know that you have that condition. But apart from that, it seems to be very safe.
Yeah, it's worth saying that if you're allergic and you have an EpiPen, as Anne says, that means your immune system is slightly more active. And of course, you're giving someone an immune booster here. So then it goes off a bit. You know, you get a bit of a boost in the allergic response. That's the way to think of it. But it's easy to treat. All those cases were manageable. You know, they were able to treat them quite quickly in the hospital setting. But it's something to keep an eye on, all right. I mean, that's why we're especially cautious now. You have to wait 15 minutes, 30 minutes if you might be allergic, just to keep an eye on you. And if allergy develops, it can be treated.
Yeah, and that's one of the challenges, isn't it, then, for people actually getting the vaccine, that you do need trained medical personnel just in case. Yeah.
And it is something that the regulators and the the HSE are following up on, as Abina will know as well, and taught me well in the last few years, you know, about pharmacovigilance and things like that, that, you know, we do need to follow up on these. They are new products.
Yeah. Absolutely. So we had a lot of questions about the different variants, but I think you guys have covered an awful lot of that in your discussion. Some other questions, I suppose, were linked to whether or not the current vaccines, because they haven't been tested on pregnant women, and a lot of the time those who are immunosuppressed, when might those populations be eligible for the vaccine? Would there be any concerns? Now, I know, Anne, you already touched on why we don't want to have those that are immunosuppressed to get to catch coronavirus or to catch COVID-19, because it gives it longer to incubate and potentially mutate. That isn't obviously the issue with pregnant women or women that are breastfeeding. Would anybody have anyone comment on that? Maybe Anne initially.
Yeah, so it's become a prominent question in the last few days. I guess, yes, it's correct. Very few, in any vaccine clinical trial I've been involved in, you don't involve pregnant women for a theoretical risk. As well as that, you do developmental toxicology studies in animal models to have a look to see if there is any risk. And that's what happened in these trials for COVID vaccines. Now, interestingly, in the Pfizer clinical trial, there were actually someone who did become pregnant, both in the placebo group and in the vaccinated group. So there isn't an issue on being able to become pregnant if you are vaccinated. It is a theoretical risk, I think, at the moment. And there are studies due to be finished and submitted to the regulator in the next, I presume, few weeks that will finally answer that. I would be really surprised. I would be shocked if there was any a risk associated with. But at the moment, you know, in the interest of erring on the side of caution, the health authorities here in Ireland, as I understand, have said that, you know, if you are interested in becoming pregnant, to wait a few weeks after you finish the vaccination. And that really is, I think, at the moment, until we have those final I's dotted and T's crossed on those other studies, it is for caution. I mean, maybe Luca or somebody else wants to comment as well.
This is true, and you might know more than me. Pregnancy is not a contraindication for any vaccine, is it? If I can use the word contraindication, that's probably the wrong word for it, but it's not an exclusionary criteria, is it?
No, but there aren't that many vaccines that we use in adults, but the ones that we do, it isn't. I mean, pregnant women are definitely indicated to get a flu vaccine. I mean, you really should be vaccinated if you're pregnant with
and the pertussis one, the whooping cough.
Exactly. So most other vaccines that we have are developed for children. Where the issue of pregnancy isn't obviously there.
Peter, you might just jump in really briefly on that, because I know just in some of the questions that were submitted, one of the concerns was the mRNA being a new technology or new to most people. That was one of the issues, I suppose, that people were raising with regard to pregnancy or breastfeeding in the vaccine.
So I don't think it would have any I would have any concern about the mRNA per se for, you know, vaccine being safe for women. It's just the way we train our immune system. Similarly, when you take the DNA molecule, or it's just the outcome, how we train this immune system, if we use the same kind of targets for vaccines, it's practically identical in the sense that all of those vaccines show good safety and efficacy. So I wouldn't say that the modality that we use would have an impact or on indication whether it should be used in pregnant women or not.
Yeah, I think, Sarah, goes back to my earlier point. The regulators are very cautious, and it's really it's a precautionary step. And I think the other side is, it's important, you know, the idea of herd immunity, it kind of got bad press lately, but with a vaccine, what you're trying to do is get herd immunity. There's some people who a vaccine might not be suitable for, and it's really important that the rest of us who can take it reduce the level of the virus in the community so those people can live safety. And we saw that this year in UCC. We had a mumps outbreak, you know, and it's because there was less people vaccinated for mumps. So we had an outbreak occurring. So I think, though, back to your question about the pregnancy and breastfeeding mothers, I think it's purely a precautionary step at this stage till more data is gathered.
Absolutely. And that's an excellent point, that we're vaccinating healthy people to protect the most vulnerable in society who may not be able to vaccinate. And that's a really good point. We have hit time, guys. I might just ask one more question that came in because I really liked it. Luke, this one was for you, and clearly from a fan. It's like a, you know, the rock star kind of a question. It was, Luke, when you get vaccinated, what vaccine would you like to receive and why?
The one I get for free. No, they're all free.
Excellent.
Well, at the
All
moment, if I look at the safety profile, and as Anne was saying, you can read it all yourself, you know, everything's available. So any of them, because they're all equally efficacious. Well, I mean, the efficacy is very high for Pfizer and Moderna,
excellent.
obviously. I'd even take the AstraZeneca vaccine without doubt, by the way. Now, there's been a few issues about that one, and mainly to do with the release of the information was a bit ham-fisted. But I'm sure Anne would agree that's a very efficacious vaccine as well. And even if it's 60 to 70% efficacious, I would take that one. So I would take any of them, at least those three at the moment. And I think, as Abina said, I mean, if the regulatory authorities approve them, you can be rest assured with them. You know, we sort of devolve our responsibility onto them. Of course we do, but they're the real experts. If they say go, I'll take any of them, you know.
Great stuff. Guys, I just have to thank you all very much for your time, and to everyone that logged in, thank you all very much for logging in, asking so many questions. I'm so sorry we didn't get to all of them. We might have to do this again sometime. It was absolutely fantastic. Thank you all very, very much. And I hope everyone enjoys and has a great BT Young Scientist.
Absolutely.
Definitely
Best
check
of luck
in.
with the BT Young Scientist now. Hope
Yeah.
it's all good fun, even though it's probably all remote, isn't it, Sarah? There isn't much.
It is, but well worth in logging on to the
Yeah,
exhibitors
exactly.
area, or particularly the student projects. I was reading through them this morning. Absolutely fabulous.
Yeah.
And past young scientist, not our participants. It's a great experience. Yeah, and well done for taking part to everybody who's involved.
Yeah, fantastic. Particularly, I can imagine the challenges we're all finding conducting research in universities at this time. So conducting research from your home, or, you know, the lack of a school lab or anything like that, it makes everything even much more impressive. So well done, everyone. And thank you all for a wonderful panel discussion.